Today we are going to study about the development and regulatory implications of polymorphism. In the last class, we had studied about the phenomena of polymorphism and what are its implications on physicochemical properties. Today, the focus would be on understanding the effect of polymorphism on pharmaceutical development and the regulatory affairs. This slide captures the drug discovery and development protocol, which we have studied earlier also. This is the drug discovery phase. This is the preclinical phase, which is followed by phase 1, phase 2, and phase 3 of clinical trials. Thereafter, the technology transfer takes place and the product is launched. The approximate time period required for these activities is given here: 3.5 years, 1 year, 2 years, and 3 years. These are the numbers of the new chemical entities which enter into the discovery phase. For every 5,000 to 10,000, sorry, 5,000 to 10,000 new chemical entities entering into the drug discovery phase, only one will reach to the market. This is the cost that is increasing as the progress in the drug development phase takes place. Our emphasis is going to be on at what stages of these activities polymorph screening is carried out. The first stage where the polymorphs become important is the preclinical phase, because in the phase 1 investigational new drug application, the regulatory agencies demand that the information about drug polymorphism in brief may be provided. The polymorph screening should be completed by the early phase 3 of the clinical trials, because thereafter discovery of a new polymorph or phase transformation to a new polymorphic form can have serious implications. So this is the latest point up to which the polymorph screening should have been completed. Polymorph screening involves generation and characterization of the polymorphic forms using various experimental protocols that are accepted in the pharmaceutical industry. Polymorphism has all kinds of concerns, intellectual property related concerns, because new polymorphic forms can be patented and it can provide a source of income to the pharmaceutical companies. Obviously, the biopharmaceutical properties of different polymorphic forms are different. That is why it is important to study them during the drug discovery and development phase. It is also a regulatory concern, which means the regulatory agencies are interested in knowing which polymorphic form has been used in the product development and how you are going to ensure that the same polymorphic form is retained throughout the developmental cycle. These are some of the aspects which we are going to discuss in more detail in today's class. The main concern during the drug discovery phase, which is the early phase before the drug development takes place, is the relative solubility and stability of different polymorphic forms. Therefore, during pre-formulation, the following issues need to be addressed, or the following questions need to be answered. How many polymorphs exist for a given API? How stable is the metastable form? This will help us in deciding whether the metastable form can also be used for the purpose of pharmaceutical development. Is there any amorphous form, which again would help us in deciding whether amorphous form can be used for pharmaceutical development, especially for poorly water-soluble drugs. Can the metastable form or the amorphous form be stabilized using formulation techniques? What is the solubility and bioavailability of each polymorph? This needs to be correlated to the BCS class of the drug. Will a more soluble form or the metastable form survive processing and storage during the pharmaceutical development? These important questions need to be answered during the drug discovery phase, as they will help us in deciding whether a metastable form or the amorphous form can be used for the purpose of pharmaceutical development later on. The ultimate aim of any regulatory guideline is to assure safety and effectiveness of a drug product. Quality is the underlying principle in both the places. Polymorphism has the capacity to affect both safety and effectiveness of the drug product. That is the reason it is a regulatory concern. Multiple solid forms can have variable physicochemical properties and biological performance. Physicochemical properties include solubility, dissolution rate, hygroscopicity, compactability, processability, and so on and so forth. The stability of the polymorphic forms can also differ from one another. FDA's drug substance guidelines state that appropriate analytical procedures should be used to detect polymorphic, hydrated, or amorphous forms of the drug substance. The guideline also says that it is applicant or the company's responsibility to control the crystal form of the drug substance, and if bioavailability is effective, to demonstrate the suitability of the control methods being adopted by the company. Control method means the analytical methods that the company relies on ensuring the identity of the polymorphic form or the amorphous form. This decision tree tells us that what and when regulators review with respect to polymorphism during the development from the NCE into the drug molecule. At the preclinical stage, it is the first time when the polymorphs might be encountered either knowingly or unknowingly. Not much is expected by the regulatory agencies at this point, except that they would like to know the identity of the polymorphic form and whether the phenomena of polymorphism has been observed at this time. During this stage, the regulatory agencies do not insist on a well-defined polymorph screening exercise because this is a very early stage in the drug discovery stage. After the phase 1 IND submission has been carried out and the clinical studies of phase 1 and 2 and 3 have been carried out, at the end of phase 2 of the clinical trials, the regulatory agencies expect that detailed data on polymorphism and effect of bulk scale-up on polymorphism is reported in the regulatory documentation. Data proving use of same polymorph in all the clinical trials should be submitted. If the company has used different polymorphic forms during the different stages of development, then they will have to demonstrate equivalency between those different polymorphic forms. Thereafter, the new drug application is submitted and reviewed. At the initiation of the phase 3, potential of the drug to form polymorph during processing and storage should be known. Appropriate specifications should be there if required to control the desired polymorphic form. Bioequivalency of polymorphs should have been established. Effect of drying temperature, milling, storage, and humidity on polymorphic transformations should be known. Possibility of solid state transition in dosage form should be studied. Kinetics of phase change should be known. Any scale-up in bulk synthesis should be done with prior knowledge of effect of the solvent on polymorphism. The marketing application, which is submitted at the end of the phase 3 of the clinical trials, should identify possible problems or the lack of them in drug synthesis and to suggest appropriate means to control the polymorphic forms. After the marketing of the drug product has been carried out and the product has entered into the post-marketing phase for clinical trials, impact of polymorphism on product quality should be clearly understood. Scale-up and post-approval changes should consider the possibility of polymorphic transition or lack of it due to composition or processing changes, and any changes in the bulk drug supplier should be done with the knowledge whether a possible change in the polymorphic form can be possible. This is the entire pathway of conversion of a new chemical entity to a drug molecule that has been marketed. And we have studied that at which stage, what are the expectations of the regulatory agencies like US FDA from the pharmaceutical company with specific reference to